So we've talked about all of the saponifiable lipids so far, the ones that contain the ester bonds in them. We're now going to take a look at the non-saponifiable lipids. And there was only one category of these, and these were known as the steroids. And steroids are characterized by having a very distinctive structural feature, which is that we refer to it as a steroid nucleus. And what it's made up of is 3 cyclohexane rings fused to a cyclopentane ring, right? And you'll see that in every single steroid, right? There's a notation in terms of how we designate the different rings and how we number them. You don't have to worry too much about the numbering of the rings and the labeling of the rings. You should be able to recognize, though, this steroid nucleus. Right? It's very hard to miss. It sort of jumps out at you when you look at a structure. All right, the most common, most important steroid in the body is a molecule known as cholesterol. All right, and the name here, the ending of it, kind of gives away what functional group is present. The OL tells you that an OH is present in the structure, and specifically there's the OH, right? You'll notice here that it does contain, it's highlighted in yellow, and I'll put a box around it in green. It does contain that steroid nucleus, right? And when you look overall at your steroids or your cholesterol and all the steroids, as well as all the other lipids we've looked at so far, is that they contain lots and lots and lots of carbons and H's, right? And that's what makes them very non-polar molecules to begin with, right? And that's how we define our lipids in the first place. All right, so cholesterol, most important steroid in the body. You get it from your diet, for example. It's also actually made in the liver. And it's needed for a variety of things. It's needed for the cell membranes. It's needed for your brain and nerve tissue, for different steroid hormones. And then it's also needed in the synthesis of vitamin D. If there's too much of it, just like when there's too much of anything, it can cause problems. Specifically, it can clog your arteries by the buildup of this thing called plaque. And as a result of that, it can lead to restricted blood flow and high blood pressure as well. Like I said, having the right amount is going to be important. Having too much is not a good thing. Having too little is also not a good thing. So I'll leave it up to you to kind of read through some of the health benefits and the cholesterol content of different types of foods. That's just interesting information. One downstream consequence of cholesterol is the formation of things known as bile salts, which are made in the liver from cholesterol, and they're stored in the gallbladder. These are going to have both polar and non-polar regions as well, as we'll see why here in a second. And they actually make fats soluble in water when they otherwise would not be. If you remember from the end of the previous section, we saw how these things called micelles would form. Well, they work in a similar type manner. These salts then will help in the absorption of your cholesterol. When there's large amounts of cholesterol accumulating in the gallbladder, then we have these things called Gall Stones that end up being produced. So here's an example of a typical bile salt. All right, this is called sodium glycolate. It's a salt, and it's a salt because it's got a charged portion associated with it. And then the majority of this, again, also you have your steroid nucleus, and there's lots of C's and lots of H's, right? So it's a salt because of that charged area or that charged region of the molecule. We had mentioned this idea of transporting lipids throughout the bloodstream briefly, and if we think about it, lipids we know are very non-polar things, and if they need to get from one part of the body to the next, they're going to have to go through the blood, right? And the blood is made up mostly of water, mostly H2O, which means that it is going to be polar. Right? This is a problem. You want to transport your non-polar lipids throughout the polar bloodstream. Well, how do we make the lipids in a way such that they will dissolve in the polar bloodstream? Well, we convert them into molecules known as lipoproteins, where we combine them with proteins to form water-soluble complexes, right? So the fact that now we're going to add some sort of regions or parts to your lipids changes their characteristic. It makes them more soluble in water, and now we're adding proteins to make them more soluble, so we call them lipoproteins, right? These are going to be the most dominant form of cholesterol in the blood, right? You're going to have these water-soluble form of cholesterol and other lipids in the bloodstream. So your lipoproteins form when your non-polar lipids get surrounded with other types of polar lipids and proteins that allow them to be transported to the cells. And what makes them water soluble, so soluble in H2O, because the surface then of them is consisted of, or is made up of, these polar lipids, right? So if we were to zoom in here on an example of a lipoprotein, right, you'll see the interior portion of it is made up of non-polar lipids. These are in the middle. And then on the surface, on the outside, right, we have the polar regions. So the polar surface. And those are made up of proteins, and you have your phospholipids, for instance, that impart some polarity that allows it then to be soluble in water. Okay, so if you think, fundamental level, what this is applying is that simple rule we talked about back when we looked at polarity of molecules and bonds, for instance. Like dissolves, like explains, and I said this in back in chapter 6, that it explains a lot about how molecules behave, and at the time you maybe didn't see the connection. Well, hopefully now you're seeing that connection, right? The fact that something is polar allows it to be soluble in a polar solvent like water, which is what you have in the blood. And by attaching polar things to your non-polar molecules like your lipids, you actually allow them to be soluble in the blood so they can be transported from one part of the body to the next. So these lipoproteins, we saw a couple different types of these in the previous example. We talked about low density lipoproteins, LDLs. We talked about high density lipoproteins. We also have a couple other categories. We have very low density lipoproteins, VLDLs, and then we also have chylomicrons. And if you look quickly here, you can actually see that what makes something high density versus low density is the relative amounts of protein that you have. All right, so high amounts of protein would make it high density. Low amounts of proteins would make it low density or even very low density. All right, that's the general trend or relationship there between those. As a reminder, also your HDLs are considered your good cholesterol or good lipids, and your LDLs are considered your bad lipids or your bad cholesterol. All right, and really that has to do with how much of each of those you have. Another place where we're going to see steroids and where they function is in the use of hormones. And you're going to talk a lot about hormones when you get into your biology classes. But for right now, what we're going to think of hormones are as some sort of a messenger, some sort of a chemical messenger that serve as a communication system for the body. So in other words, they transmit messages. So they're released in one location, they go to a different location, and they have some sort of an effect. You can think of them as, sort of like a FedEx delivery person or a UPS delivery person, right? Takes a package from one place to another place, and then it delivers it, and it has some sort of an effect where it gets to. Your steroid hormones are made from cholesterol. They include your male sex hormones, things like testosterone, etc. You have your female sex hormones as well, and then you have some other types of molecules known as your corticosteroids made from the adrenal glands, which help in balancing your electrolyte levels, but also regulating blood sugar levels, your blood glucose levels. All right, so we'll take a quick look at some of these. And again, when you look at their structures, you'll see that they all share something in common, right? And that's their steroid nucleus, right? All of these are going to look really, really similar to one another, all of them. What makes them different really is just by tweaking different regions of the molecules. So for example, if you take a look at a male hormone like testosterone and you compare it to a female hormone like progesterone, all right, what's the difference between these two? Right, if you look closely, the only difference is in this region here. Right, everything else is the same. So in testosterone, you have an alcohol group. In progesterone, you have a ketone group. That's the only difference between those two, and maybe an extra carbon or two. But that simple difference has a huge difference in the effect or in the function of those two molecules. So as you read through the remaining part of this subsection, I'm going to leave this up to you, where they talk about the chemistry linked to health. There's some really good information in there. It talks about anabolic steroids, etc. Like I said, it's a really good information. It kind of shows you how you can apply some of what we learned in the immediately preceding section to maybe some everyday occurrences. So take a look at that, and then we have one more section to talk about, which is the cell membranes.